All right, shall we begin?
Yes.
Good morning, everyone. This is Jack Van Horn from the BD2K Training Coordinating Center based at the University of Southern California, and I'd like to welcome you all to our next installment of our BD2K Guide to the Fundamentals of Data Science seminar series. This morning, we're absolutely delighted to have Pascale Gaudet from the Swiss Institute of Bioinformatics, who will be lecturing on data curation and version control. Dr. Gaudet has been working as a biocurator since 2003, first with the Model Organism Database, dictyBase at Northwestern University in Chicago, during which time she became heavily involved in the Gene Ontology project. Since October of 2010, Dr. Gaudet has been the scientific manager of the neXtProt database at the Swiss Institute of Bioinformatics, which is based in Switzerland. She is one of the founding members of the International Society for Biocurators and has served as its chairperson from 2009 to 2013. This morning she's going to be again talking about data curation and version control, and she will attempt to explain what a biocurator is, what biocuration is, and what is the process by which the life cycle of scientific data goes through, and discuss relevant issues. For all of you who are listening, I encourage you to chat in using the little chat box feature of your questions, and I will act to kind of curate them myself and ask them on your behalf. Without further ado, Dr. Gaudet, welcome and thank you very much.
Thank you. Introduction. Thank you for inviting me to give this lecture. I'm really... Can you see my screen okay?
Yes, we can.
Okay. So just to say, it's a big honor for me to be part of this course, and I'm very pleased of the recognition by BD2K of the importance of biocuration in data science and biomedical research. So this is to summarize what Jack just said about me. So I'm the scientific manager of neXtProt at the Swiss Institute of Bioinformatics. I'm also still currently a manager on the Gene Ontology project, specifically on the phylogenetic annotation approach project. And here I copied what some other previous presenters did, and I gave people my email and my Twitter account in case you want to continue discussing what I'm going to be talking about today after this talk. So databases right now are a cornerstone for research. Curated databases are a tool that most researchers right now on a daily and many users spend several hours per week on various databases. When I was at dictyBase, we surveyed our users, and many people estimated that they were spending probably 1 day a week overall on various resources. However, database creation and maintenance is an obscure process to most users. Sorry, I just need to get rid of this. Okay. Yeah, so database creation and maintenance is an obscure process to most users, and this is what I want to talk about to you today. So I'm sure everybody here has interacted with many of these different types of resources, but this is to give you an idea of, or to remind you of the scope of curated resources in the life sciences. So there's databases that talk about protein functions, genomes, chemicals, clinical trials, interactions with ligands, with drugs, with proteins. There's a large number of databases. In fact, the main reference in terms of biological databases is the yearly nucleic acids research database, which in 2016 listed 1,665 different databases. And there's the link here for the list of databases that are compiled by Michael Galperin, who's the editor for the nucleic acids research database issue. I want to point out that this is not an exhaustive list of all the databases that exist. The NAR database issue selects resources that are deemed of high quality, that are non-redundant with other existing resources. So in other words, if you want to propose a repackaging of other databases, it's usually not going to make it in the collection of databases. And also, they select resources that are maintained. So just to say, there's a large number of what we call niche databases, usually created for specific projects, but that are not maintained once, for example, the students or postdoc moves on to a different project. So these are excluded from the nucleic acids research database issue. Even within this collection, there's still a small number that goes obsolete every year. And so just as a point to mention, this year there were 23 resources that were removed because they didn't seem to be maintained anymore. So today I'm going to talk about what is biocuration, why do biocuration, and how do we do biocuration. So to give a definition of biocuration, we can say that it's the analysis, interpretation, and integration of biological information into data repositories. So it's in part data capture, but that's really just one aspect. To do biocuration, you need to understand the data, you need to analyze it, and you need to distill what the researchers are trying to show in order to translate this into a language that a computer will be able to understand. And this is a little subtlety, without falling into the trap of over-interpreting data when, for example, interesting new theories are discussed in papers, but there's not the exact supporting evidence that one needs to really say that this is an observation that's been shown, whereas this is a conclusion that we might want to test in the future. So this data is integrated into some structured form. Usually it's a database, but it doesn't really have to be. This is very technical. I'm not going to focus on the technical aspects. And so really biocuration encompasses a wide range of activities. So this definition may not include all the different activities, but to give you an idea of some of the typical tasks that a biocurator might do, it's to conceptualize certain types of data, to create databases, to evaluate data, insert it in the database, and also, if possible, link it so that it makes sense biologically, and obviously store it. So why do biocuration? I mean, right now, as we know, modern research generates huge quantities of data. And while each data set is very rich, or each data point even, is very rich on its own, where it becomes powerful and interesting is when we can bring the different data from different groups, from different types of experiments, and so on. And so one aim of biocuration and curated databases is to be able to connect and query all the knowledge that we have generated as a scientific community. I mean, I think this is anyways a common goal of the entire scientific community. So why biocuration is also because papers in the traditional form are not easily amenable to computational analyses. So the data that is there, the knowledge that is there, is very difficult to extract. So it used to be that when there was a little less literature accumulated, people could be expert and have read everything in their field. But more and more, this is becoming almost impossible. Another thing is that biological data is very heterogeneous. So there's many different techniques that ask similar questions. New techniques are constantly being developed. So one of the challenges is also systems where all this data can be a little bit more interoperable. And so this is not exactly built in into how people currently do experiments. So right now we're at a stage where the data and the knowledge generated by experiments cannot be immediately consumed for other types of analyses. So right now the process is to convert this into a form that can be computed. So the idea with biocuration is that if we have data that is correctly captured, then we can facilitate retrieval, extraction, indexation, and make the data as a whole more valuable. So what we're hoping for that a curated knowledge database will provide is easy access to data that is, so I'm repeating this a little bit, but this is really very important in terms of why we're doing state this. Also, and ideally the other thing is it enables further analysis. And just a point on the current, sometimes if you haven't read other articles that have been published afterwards, you don't know if what was demonstrated in that article is still the best you're looking at. So ideally we would have a way, databases aim to provide the current state of knowledge. So data that has been shown to be incorrect or inaccurate should be removed, corrected. Okay, so how do we do biocuration? There is really the first one is manual and the other one is automated or assisted with text mining. I'm going to describe both approaches. But first I'm going to focus on the manual approach. So some of the, so I just want to say that for today's talk, I give some examples of literature curation. Obviously, there's many types of data that are curated. There's many types of databases, like I mentioned earlier. So for the examples today, I'm going to focus on literature curation, just for simplicity. Unfortunately, there's no time to go over all the different types of curation that can be done, but one that comes to mind, really very important thing that I won't be talking about is genome annotation, gene models, sequence features, and so on. So here's an overview of what a curator does as specific steps when annotating a paper from the literature. So first, you need what I call bioentities are in the paper, because usually what people want to do is to connect a modification or a phenotype. So you need to know what gene is being described in the paper. It seems trivial to people doing research and writing papers, but when you're on the other end reading papers and trying to extract this information, in fact it's an extremely challenging task. First, there's trivial. For example, when a gene name is the synonym of another gene, so sometimes you need to a paper is talking about issues of that can be people describe variants or even post-translational modification. Amino acid position on the current reference sequence is different from the one that was presented in the paper. It needs also to be resolved. And also, it can be difficult to find the species that is described in a paper. There's several cases that we have encountered where a paper talks about a human process, but somehow when you read in very much detail the materials and methods, or sometimes even it has happened that we had to do a blast and then we realized that in fact there was a mouse protein being expressed in the human cell. Okay, so we just always want to make sure that whatever information is extracted from the paper is linked to the correct biological entity. So this is the first thing, is to just extract which gene the paper or genes the paper is talking about. Also, obviously, you want to extract whatever real information, knowledge is published in the paper. So, for example, they show a molecular function, or they show a pathway in which the gene is involved in, a phosphorylation site, a phenotype, and so on. And synthesize the information to represent, like I said, the current state of knowledge. So it also happens that this occurs at the level of data presentation. There are some examples of bioentity recognition issues. So maybe some of you are familiar with APC, which represents either a protein involved in colon cancer or the anaphase promoting complex. So people familiar with that literature would immediately know that these are different kinds of papers, but it's not trivial for everybody to recognize. There's also here I put some Drosophila genes, but it's not that it's the only case where there's genes that are named with words that are real English words. And so obviously this can also cause false negative problems. You're looking for the literature for that gene, and you find a lot of papers that talk about white, but it's very difficult to find the papers that talk about the gene white. And I mentioned in the previous slide the sequence isoform species issues, but this is something that curators spend a long time trying to sort out when annotating a paper. So to discuss a little bit the second step of the biocuration task, I'm going to give you an example of work that we've done in my group. So we've curated the functional impact of variations in the human genome for a number of cancer variants. And so we were interested in really capturing the molecular mechanism of these variants. So we captured enzymatic cellular processes, in molecular binding, changes in protein or in subcellular localization. And this is the overview of the end. I'm sure many of you have seen these types of databases where you can go and browse some information. Show you the phenotypes of the gene MLH1 involved in colorectal cancer. And at the first level, we show the different phenotypes that are tested for this gene. So there is, I cannot get my mouse to work here, but we have divided them by groups. So the molecular function, the molecular function, the ATPase activity. There is also impact on biological process, so this match repair. Its localization, which is nuclear, and impact on some important binary interactions. And you can go in and search for any gene that has a defect in a specific phenotype you're interested in. So for example, you can look at the list of variants that have defects in mismatch repair. And so here there's 4 different variants listed. So this is a partial 100 different variants for this gene with problems in mismatch repair. And so we also show all the evidence for the data that we're capturing. So this is, I think, very important in terms of trusting the data. Anybody who downloads this list can then go back to the original literature and decide if you agree with the information which is presented there. So, so this was the end result. So going back on how we actually extract the information. So this is one of the articles that we used to annotate MLH1. And so one of the challenges is dispersed. In this case, there's information dispersed within the same table, but sometimes it's even more widespread in the paper. So we have the gene name, MLH1, part of the title of the table, a list of mutation in the table. We also have the phenotypes in which we're interested in that I highlighted in yellow. So the MSI status, it stands for microsatellite instability status. So this is something that tumors, and it measures essentially whether or not the mismatch repair system is functional or not. And in this case, it's given a qualitative value, high, low, or not tested. And then the other column we were interested in capturing, like I mentioned, is the protein stability. And so in this case, it's tested by immunocytochemistry. So we know this because we know that in this case, immunocytochemistry measures the level of the protein in the cell, but obviously you can do immunocytochemistry to measure many other things, just the localization and so on. Also, there is in this table a number of variants that are not original from this paper. So for some reason they thought these were interesting and they added them, the authors added them in their analysis, but the data was, and so in these cases we don't information just cited because we only capture each experiment only once. If it's cited in a secondary publication, we don't capture it. So this, because otherwise it would give artificial weight to the assertion because it's been found X number of times. So I think it's very important that anytime something has been shown, that data doesn't get duplicated in the annotation process. This is a similar data, a different paper. So in this case, they also test the histochemistry, as you see on the left, and they tested in what they call step 2 MMR assay, which is also testing mismatch repair. It's very similar data, it's just presented in a very different way. So the legend says that the black cells represent pathogenic or deficient, white cells are non-pathogenic. The diagonal line means that it hasn't been tested, and the gray cells mean that there is conflicting data. So this is something that manually we can understand, but that automatic annotation systems, I don't know of any system that can extract the information from such a table. Now, this paper that you find quite frequently in research papers is a small case F next to the variant PK618 alanine. So in the legend it says that these families have additional mutations. So in this case there's 2 mutations in the same gene. And so of course footnote may be very important in terms of the explanation of the phenotype. So in this case, we're looking at a double mutant, but in the table, it's presented as a single mutant. So again, this is some of the very detailed work that our team and many other teams does to make sure that we extract all the information accurately. Now, I mentioned that There's a microsatellite instability, and the second paper had MMR, which is a mismatch repair assay. There's different types of experiments that test the same thing in this mismatch repair field. So in this case, I listed 4 different experiments: microsatellite instability, in vitro mismatch repair assay, mutator phenotype assay, damage. These assays all reflect defective mismatch repair pathway. So we believe that it's very important that these be captured in the same manner, or there's 4 different. To explain a little bit more about the work on gene ontology, I think many of you must be familiar with the Gene Ontology. It's one of the most widely used bioinformatics resource. It's a shared language for the annotation of molecular functions and biological process and cellular components across different organisms. And it covers 3 different domains, like I just said: function, process, component. And develop this is exactly to be able to standardize the description of gene function. Like I just said, biocurators link gene or gene products or modify proteins and so on to terms of the gene ontology or various ontologies in order for the data to be organized in a consistent way and for users to be able to consume it in a way that is. So in terms of the MLH1 protein, for example, that I was mentioning, its function is to bind another complex. It's involved in mismatch repair, and it's localized to the nucleus. Okay, so, and it has a number of other more subtle gene ontology functions, annotations. Gene ontology annotations to function or process and component, but these are the most central ones. So I suppose that you might be familiar with other biomedical ontologies. There's right now biomedical ontologies in most areas, so to describe anatomies, chemicals, diseases, experiments. So the mass spectrometry community has an ontology that describes the different techniques that they use. There's also, in the Go, you can describe protein function and subcellular component, can describe phenotypes, can describe proteins and protein families, sequence, and so on. So this is really one way to represent the data that also has been proven to the research community if we just look at how much the gene ontology is being used in bioinformatics applications. So one comment that we often get about biocuration, manual biocuration, is that it's long and it doesn't scale easily. Although it's very accurate, we need to develop ways to make manual annotation. So I'm going to give you one example in that direction. In the Gene Ontology Consortium, we have developed a tool called PAINT, phylogenetic-based annotation inference. And what we do is we make gene ontology annotations relations. So I'm just going to spend a couple of slides describing this. Here what we have in on the left is a gene family for MSH2, which is another member of the mismatch repair complex. So there's proteins, this is conserved from human to E. coli. And so there, if the sequences in green are experiments made by other groups, and we can do using this is we look at the relationships of all these genes, and then we can decide where a function first evolved. And then we, what we call propagate or infer that function to a node, a phylogenetic ancestor that we choose, and is the second step of the annotation, where from this ancestor the annotations are propagated to all the ones in blue, the ones that didn't have any annotations. Okay, so the workflow is to look at the experimental annotations shown here in blue, and so essentially the process is we look for the most recent common ancestor, and then we decide if we want to annotate to that, or if we think that it should be annotated to a more recent ancestor or a more ancient ancestor. And so the information, these decisions include, for example, sequence conservation, active site presence or absence, and a number of other things. And so I don't know if you can see this alignment, probably, but we can also annotate lots of functions. So it happens in big multigene families that certain functions are lost. In this case, it's because an active site is lost. The cysteine is of PGM5, but in the PGM1 clade, the cysteine is mutated to an asparagine, and therefore the function is lost after this duplication event indicated in the green circle. And so using this to say, that there has been a loss of function. And so this is to give you some idea of how much more we can do using this more efficient curation system. So as of last year, we had 2,500 different families curated. So the families are based on PantherDB. Right now, there's a families in the whole PantherDB set. So we've done about a quarter. And within those 2,500 families, there has been over 5,000 human genes annotated and 20,000 new human gene annotations. And if you look at also all the organisms, we have nearly 1 million annotations with this system. This scales quite well in the sense that one curator can do about 1,000 families a year, which, if you take the average, it corresponds more or less to 100,000 genes. So the only drawback is that, of course, we still need the primary annotations in order to be able to do this. But the nice thing is it annotates multiple new no annotations. So now I'm going to switch gears a little bit to talk about automated curation. So the manual curation is very nice, but like I said, it's time consuming. And there's a lot of hope that some automated systems could help us do curation more effectively. So the task that automated curation tools that I'm aware of do include document retrieval, document prioritization, what they call, right, so let's say if you're interested in a specific gene, a specific disease, it can retrieve documents that are relevant to that gene disease process. It can, these tools, these documents, which ones are more likely to be more interesting to you. Gene normalization task is to try to identify which gene the paper is talking about. So this task, as far as I know, maybe there's been more recent developments where this is, as I explained earlier in my talk, even manually we have a lot of issues. So the automatic systems were not usually performing really well in this task. And finally, another task is concept identification. So which gene is mentioned in the paper or is mentioned in the paper, and so on. So these are all tasks that we can ask automated systems to help us with. One very popular tool in the biocuration community for automatic annotation is called Textpresso. It's developed by the group who also is PI of the Worm-based database. And so it was first implemented in Worm-based, but it's now and more frequently being implemented in other databases, dictyBase and TER, the Arabidopsis thaliana resource. So what Textpresso can do is, if it's given on the left a number of PDFs of relevant literature, gene product names and synonym that it can use as a search base, and well, for TER, some plant cellular component terms. So essentially you have your literature and your list of genes and interested in. What Textpresso does is it converts the PDF to text. It tries to identify the gene product names. It marks up the text of the paper. It indexes it. And then it presents it to the curator in a way. So I'm just going to show you. This is from the 2012 papers that describe implementation of Textpresso in dictyBase and so this is the curation interface. So at the top is the title and abstract of the paper, and then at the bottom you see the gene protein name that the system extracted, Gene Ontology cellular component term, nuclear, that it found in this. And then you can also select more specific or less specific terms. And then the curator just sees this pre-processed annotation and can hit make the curation, make the annotation or don't make the annotation. And this goes directly in the database. Now, in automatic annotation, the gold standard measures are precision and recall, are the fraction of the documents retrieved that are relevant to your query. Okay, so how much of your corpus did you really find? And the precision is the fraction of the documents that are retrieved and that are relevant, because the system obviously can also give you some false positives, right? So the recall is, it measures more or less the false negatives versus the false positives. And so this is again from the paper. So for dictybase and TER, the recall was maximum, so there's still 50% of the literature that the curators needed to go back to and annotate manually. And the precision was between 60 and 75%. So sometimes there were a number of false positives too. But despite that, here I call this a cost reduction. That's not how they phrased it in the paper, but in the paper they say a 2.5-fold increase in curation efficiency. So it's still pretty good. You can annotate more than 2 times the number of papers that you would without the system. The cons here, I would say that usually these things don't take in the cost, don't take into development, maintenance of the infrastructure that you need behind. And also there's large projects in this area that to a real life setting. Okay, so it's right now it's still pretty embryonic, and there's lots of research going on, but in able to use automated methods for biocuration, there's a lot of tasks that cannot be done. Okay, so we talked about what, why, how, and now I would like to talk about who does biocuration. So as Jack mentioned before, biocuration is done by professional biocurators such as me. So we surveyed the biocurators back in 2012, and there's a paper that described who are these people and where they come from and why they became interested in this area. But just to say that most people have a lot of experience. Most are PhDs. Most have a lot of experience as bench scientists, so most, and also postdocs. Some people have done some programming, and usually people stay in biocuration for a long time. So people are experienced researchers and experienced curators. Also, it was mentioned that the biocurators have a professional society called the International Society for Biocuration, and different members that represent about 150 different institutions. There's annual international meetings that are very interesting in terms of giving people new ideas and new ways to do things and to collaborate. So I think these are really, really good for the community. And it's amazing that next year is already going to be the 10th annual meeting of the International Society for Biocuration. So this is a bit restating what I said at the beginning in terms of the importance of curated resources, but with from the point of view of the biocurators themselves. So what we would like to have is to have compatible infrastructure for data deposition, exchange, and analysis. So it's important that we have common control vocabularies, compatible tools, compatible identifiers, data formats, systems, and so on. At the same time, it's balance stability and innovation. We don't want to be in a situation where standards are so rigid that people cannot start doing new types of experiments. So this is also very challenging to be back compatible, move forward at the same time. We would like for the resources that we're developing to be sustainable, and keen to get researchers involved. So that's one aspect of the one group of people who do biocuration. There's one approach that has been used. So the crowdsourcing can be divided into 3 different aspects. One is asking scientists to contribute to your research. There's Wikigenes. I know back 7-10 years ago, most of the Model Organism Databases implemented their own wiki to try to get researchers to add their own data if they wanted. RFAM is the best example of a very successful. The second approach in crowdsourcing is what people might know as Amazon Turks. So you can go to Amazon and propose, I mean that's one way, but you propose a very small task and people are paid a small amount to do 1 unit of example to identify variants in an abstract. So just to tell you, is this string of text a variant, yes or no? A paper that Lynette Hirschman published about this. It was quite successful for that small task. But then, of course, if you want to do more complicated tasks, then you need to start making bigger payments. And you also where you control for the contributors, their expertise, how many false positives they give you, and so on, like what's the quality of their work. There's also an interesting idea in developing games that could help somehow integrate data in a more structured way. So well, Foldit is not exactly going in databases, but for protein predictions was the most successful game in biology, where people, biologists and other people, could try to find the best structure for a protein of whose structure was not known. There's also Cancer Research UK, who developed a few games, and well, other groups, Andrew Su among other people. I don't know why I forgot to put his name there, but so there's a few ideas of getting people involved in a fun way, but it hasn't been picked up. It's not like everybody's playing these bioinformatics game while they're waiting for the bus or something, at least not as far as I know. So the pros of crowdsourcing are a lot of potential contributors. There's a significant cost to developing all this infrastructure. Many of them, if they're not used very much, well, it becomes money that was invested for not much return. And mostly, at least the reports I have looked at, describe pretty low rates of participation. So that's too bad, really, because it would be really great. A group which is very important for biocuration as a whole is researcher. Many research projects involve some curation, and that data is not always contributed back to databases, or often it's not. There are some journals that encourage researchers to provide data in formats that can be directly inserted in databases. So for example, Gene Ontology Annotations, a few journals request that. But clearly, the incentives are not yet there for people to really provide a lot of data. Researchers have a lot of other, there's not much recognition for having provided your data or not. So we really need to work on providing better incentives for researchers to curate their own data. And I mean, one obvious way that I can think is, and that's discussed a lot lately, it's not my idea, is to give some, either via your ORCID, your ORCID, or some other mechanism, that curated data would receive some citable unit. Okay, so the last little bit of the talk, I'm going to discuss versioning issues. So because all these curated databases are available out there, there's more and more experiments that rely on these resources for data analysis, especially in the high throughput area. However, most papers that I read that do bioinformatics analysis do not provide enough data to reproduce their experiment, their data analysis experiment. Okay, so that's one issue. This is a slightly different issue. Is I mentioned that biocurated resources want to be up to date with respect to the current knowledge. So this is an example of a case where we have a protein that is a dimer, and when the A subunit is bound to the B subunit, the A subunit inhibits the activity of the B. Now, there's a protease that comes in and that degrades the A subunit, and the B subunit becomes active. Okay, so this is not a very complicated mechanism of regulation. So the A subunit is involved in regulation. The B subunit has it's a nuclease, right? It has the deoxyribonuclease activity. Now, the reason why I'm saying this is that they didn't know that it was a complex. They had only identified the A subunit, and they thought that the A subunit had the activity. So when you read the further literature, then you realize that the resolution of the first assays were not good enough, and the results were, I guess you can say wrong, but it was just misleading. That the way they did the experiment, they didn't have the resolution to determine whether that there were two subunits there. So the idea is that to remove this annotation on the incorrect, to remove the incorrect annotation to make sure that the knowledge is kept up to date. And where I want to go with this is just an example, like a very specific example, but I want to show you that annotations evolved. Annotation evolved quite a bit, in fact. So here there's a tool developed by Paul Pavlidis at the University of British Columbia, where you can query either a gene or a gene ontology term and see how it changes over time. And in this case, I just picked protein kinase activity, and we see for different species the number of annotations over time since 2002. A lot of annotations anyways, only human annotated protein kinase activity. But it's not, as one might expect, a linear increase. There's also time where there is a big drop or a big increase. And so that means that depending on when you do your bioinformatics experiment using this data, your results can change quite dramatically. And to illustrate this, there was a paper published just a few that shows that the impact of outdated gene annotation on pathway enrichment analysis. So for high throughput experiment, pathway enrichment analysis is really something that people almost always present in the paper. And what they've shown here is they looked at a number of different articles, and they also took, they took annotations from 2010 and 2016 and checked from these different data sets which terms were most enriched. And so the purple dots are 2016 and blue dots are yellow dots are 2010, and blue dots are the ones that are found in both. So you see that there's a number of terms between 2010 and now. And you also see with the purple dot that there's many terms that are now enriched that didn't used to be enriched if you use the 2010 Go data. So it's very important to know which version of the tool, which version of the data that you're using. And so here is just a few best practices for bioinformatics experiments should be. People who use curated resources and associated tools should include publication dates and version number of their tools. So the date of the datasets and version number of their tools. And also, I find sometimes in papers people make additional annotations because they found it was incomplete. And so this information, these additional annotations are not always publicly available. So I would just like to conclude by saying that biocuration should be an integral part of the data life cycle. And so right now, it's often that the curation occurs post-publication. But I'm really hoping that we can get to a point where the biocuration will also be part of the data management plan, part of the collection plan, and part of the analysis, and that it won't just be curated after the fact. So I'm going to stop here. I took quite a bit of time, and I would like to thank you for being here and acknowledge the main projects I'm involved with.
Thank you very much, Pascale. That was fantastic. Rich talk. We've had a number of questions. We have a couple, look at a couple of the themes of the questions. They seem to revolve around quality control and how automated quality control and verification there. Also, when you're doing a manual curation, how do you decide which studies to include? And is there a panel of experts or some sort of expert? You address some of that, perhaps?
Sure. So in terms of, I think it really depends on the type of data you're looking at. So for example, for sequence information, you can certainly do that by automatically making sure that the TM you're annotating really corresponds to a position in the reference genome, for example. In terms of literature curation, it's very hard to have nets that will catch problems, but we certainly have seen papers by, for example, Christophe Desimo's group, where they compare the gene ontology annotations made predicted electronically, and then they compare how often these were confirmed by experimental annotations later. So that also helps to do some what we call sanity checks. But it's true that the quality controls are very important.
One of the other questions were, are many of the databases that you mentioned, are they publicly available?
Yeah, they should all be.
Fantastic.
Yeah.
One question I had is, you mentioned a little people who are biocurators, and I'm curious what sort of involvement you see from people from the library sciences, or almost like museum sciences, who've had some experience with this, and are you seeing some sort of comparisons between the type of work being done in libraries and museums with the type of work that's being done with biocuration?
Yeah, it's funny because I was reading articles about this recently, and my experience, and it's consistent with what I was reading, is it's not sufficient. It's true that sciences, we started to develop tools and infrastructure independently, really with the life sciences in mind, but definitely we can acknowledge that librarian scientists have accumulated over the years. So this is also another direction that this community should take, in my opinion.
Yeah, I mean, it seems like, and somebody had asked a question about a fundamental difference between being an archivist, stockpiling examples of data, versus curating that data, which is sort of an ongoing process of continuing to go back and make sure that it's accurate and up to date, as you say, versioned or dated, so that people know how to use it. Would you mind commenting just a little bit about that distinction of why that's so important?
About why the accumulation versus the well,
exactly,
I think this is where the idea is we need to know what we know right now. I think this is extremely important, and this is also as much hopeful as I am in terms of the automated methods. The automated text mining can go through all the literature and all the data sets and find relations, but they will not be able to find what has been proven inaccurate. This is what we currently know. It's really important, especially more in have knowing what we know, and it's a very high goal, but it's very important, in my opinion, to be in order to be able to progress further. There was a paper that dates from a few years by Gene Ontology that says why most published research findings are false, and that's probably an exaggeration, but there's certainly a lot of hype in the literature and a lot of things that never get reproduced once they're published once, you know. So it's great, but if you have a real current state of knowledge base, I think it's very difficult for us as a research community to make progress in making new relevant discoveries.
Well, I think, and on that comment, this is a great place to conclude our lecture from this morning. I'd like to thank Pascale Gaudet once again for a very interesting lecture, and want to encourage you all to tune in again next week on this same channel, same time, to hear the next in our series. Again, we're very excited to be bringing this to you. Please let others know. We're very excited to have a rich audience, and very excited to be doing this. Again, all the videos will be shown on the BD2K Training Coordinating Center website here within moments after the lecture. Dr. Gaudet is willing, and we'll make those available as well.
Yes.
So anyway, once again, fun and enjoy your weekends.
Thank you.
Thank you again, Pascale.